start with the next one. So genetic drift. How would we define genetic drift from what we have here? Anyone have an idea? Oh, are you guys not able to see it? Let me try this again. Okay. Okay, is everyone able to see the homework now? Or are we still having some technical issues? Okay, okay, cool. Okay, so with genetic drift, how do we define genetic drift? Yes, Jackie, so it's the effect of random chance on a gene pool, right? So if we're looking at genetic drift and we're looking at the effect of random chance, does genetic drift have a larger effect on small populations or on large populations? Smaller, right? Exactly. So genetic drift is going to be much more potent on small populations than it will be on large populations. Perfect. Okay, next one, sympatric speciation. How do we define sympatric speciation? The third to the last one, yeah, so speciation in the same geographic space, exactly. So speciation that happens inside the same geographical area, and that would be considered sympatric speciation. Perfect. Cool. Next one is population genetics. What is population genetics? All right. Anyone have an idea? So it's not the variety of alleles in the population. Kind of close though. Yeah, so it's the study of how selective forces change allele frequencies in a population over time. Right. So when we're acting as population geneticists, we want to be able to look at what's happening inside of our population, determine what type of selective forces at work in order to change all the allele frequencies in that particular population over a period of time. Perfect. All right, good. So next one is variation. Okay. So what is variation? Yeah, so this is going to be the variety of alleles in a population, right? So over a period of, or over your population, you're going to have various different alleles across individuals. Each one is going to have its own variety of alleles that will give it its particular traits. Okay? So variation is just the variety of the alleles present in the population. Perfect. Okay, what about natural selection? Yes, good job, Alan. So this is the relative survival and reproduction. Good job, Josie, too. Yeah, relative survival and reproduction of individuals in a population based on the favorable traits that they may have or non-favorable ones that they may have. Okay. And that will ultimately lead to evolutionary change. Perfect. Okay, good. Next one is a bottleneck effect. What is a bottleneck effect? Yeah, so this is a magnification of genetic drift. And so this is going to be the result of different types of natural events or catastrophes, or even some man-made events that can take place, and they wipe out large portions of the population, which significantly reduces the genetic variety inside that population. Yeah. Okay. Next one is evolution. Okay, so how do we describe evolution? Evolution, what do we think? Evolution, yeah. So this is a change in the population over time. Perfect. Cool. All right, then the last one we have vicariance. What is vicariance? Right, so this is a type of allopatric speciation that occurs whenever you have individuals getting divided up into separate groups, usually by some geographical barrier that's popped up. As they separate them, they're going to be exposed to different selective forces, and as a result, that's going to eventually lead to the evolution of those two species. Perfect. Okay. Any questions on these definitions here? Okay, cool. All right, so we're going to go ahead and jump forward. So the first question, or question 2, which of the following would lead or would increase the effect of allopatric speciation? Okay, so we have A, longer distances between divided groups, B, lower mutation rates, C, equivalent numbers of individuals in each new population, or D, higher mutation rates. Which one would increase the effect of allopatric speciation? Yes, so Uriel says A and Kimberly says A as well, so longer distances between our groups, and that would be true. So remember, allopatric speciation is speciation that occurs whenever you have individuals in two different environments, two different locations. Okay. So the longer the distance, the more different the environmental factors are going to be. If you have different environmental factors, that's going to lead to different selective pressures and ultimately lead to speciation occurring. All right, good. Number 3. So which of the following is an example of natural selection? Okay, so we have A, in a species of lizards, two types of lizards exist. Okay, type A, lizards can camouflage into their surroundings, while type B lizards cannot camouflage and are easily visible to predator hawks. Hawks eat the type B lizards until the majority of the population consists of type A lizards. Okay, that's A. B says an ice age occurs that kills a large portion of the elephant populations. As a result, most of the surviving elephants have little genetic variation. C says two populations of wolves live on opposite sides of the mountains. Over time, the western wolves leave their area of the mountains and move to the eastern side to coexist with the eastern wolves. And then D, in a population of moths, brown moths only mate with other brown moths. Okay. Which one is an example of natural selection? Yes, so Kimberly says A, and that is true, right? So good job. So this is going to be something that's going to affect their survival. Okay. If we were to look at B, if we have different elephants here that get killed by an ice age, this would be an example of a bottleneck effect, a type of genetic drift. If we look at C, we're looking at migrations where we have western wolves moving into eastern wolves, and then the population of moths only mating with brown moths only mating with brown moths. That gives us non-random mating and possibly sexual selection too. All right, number 4. So here we have a skeleton of a whale. Okay, so whales have small hind leg bones, indicated by C in the diagram. What best describes the hind bones or the hind leg bones seen in a whale? Okay, so we have A, vestigial structures that had a function in an ancestor. B, homologous structures to the wings of butterflies. C, fossil structures that exist or from an extinct ancestor. Or D, analogous structures to the fins of living fish. Okay. What do the hind leg bones indicate in a whale? Right, so this would be A. So these would be vestigial structures. Okay. So remember, vestigial structures do not have any current function. Okay, they don't have any particular function, but they continue to exist. Usually because there was likely a common ancestor that needed that particular structure. Okay. As it eventually evolved into this structure or this organism, it no longer needed that particular structure, but it continues to exist. Perfect. Okay, number 5. Which of the following is true about the theory of evolution? Okay, so we have A, organism populations can change physically over long periods of time. Okay, B, evolutions can be seen in individual organisms. C, individuals with favorable adaptations and characteristics reproduce at a higher rate. And D, individual organisms can change by acquired characteristics from parents. Right. Exactly. So organism populations can change physically over long periods of time. Exactly. Good. All right. Any questions so far on any of these ones here? No. Okay. Okay, number 6. So 6 says, which of the following is true of Hardy-Weinberg equilibrium? Okay, so A, Hardy-Weinberg equilibrium determines allele and genotype frequencies, assuming no evolution is occurring. B, it assumes if evolution is occurring. C, it says Hardy-Weinberg equilibrium is not violated if genetic drift is occurring. Or D, none of the above. Which one is true? Yes, A is true. It assumes that no evolution is taking place. Okay. So Hardy-Weinberg equilibrium will give you specific numbers or specific calculations for different allele frequencies and genotype frequencies, assuming no evolution is taking place. But then if you actually go out into the population and you look at the allele frequencies and the genotype frequencies, and if you can tell that those numbers are different from what you calculated with Hardy-Weinberg, okay, then that is an indication that evolution is actually taking place. Okay. And then you can go and figure out which type of factor is at work. Yeah. Okay. Question 7. So which is true of sexual selection? Okay. So we have sexual selection improves reproductive ability, but can be detrimental for survival. We have male competition is one type of sexual selection, and females choosing mates based on specific characteristics is an example of sexual selection, or all of the above. Which one is true? Right, all of the above. So these are all true about sexual selection, right? So it is going to improve reproductive ability, but it can be detrimental for survival, like we saw with male-male competition, like we saw with the peacocks with the bright feathers and things like that. Okay. And then the females are obviously going to be choosing mates based off of certain characteristics too. Cool. All right, number 8. So a genetic mutation causes a newt to have faster reflexes. Okay? After many generations, most of the newt populations has the reflex mutation. Which of the following most likely caused this change? Okay, so we have A, newts with the mutation are better able to survive and reproduce than newts without the mutation. B, the mutation was contagious. C, the newt passed on its mutated gene to other adults. Or D, other newts learn to copy the strategies of the mutated. Okay. So Kimberly says A, and that is true, right? So newts that have the mutation would be better able to survive and reproduce than the ones that don't have the mutation. Okay, so as they continue to survive, they could start producing more babies that also have that same mutation until after many generations, they all have the same fast reflex mutation. B cannot be correct because mutations are not contagious. You cannot pass one mutation from one adult to another. Okay, and that also kind of goes for C as well. And then D, it can't be true because newts are not going to be able to just copy, a mutated gene or a mutated strategy. It's all going to be based on whatever's in their own DNA. Cool. All right, good. Number 9. So a population of birds undergoes adaptive radiation as the founder species spreads to new habitats in the north, south, east, and west. Which of the following is a selection pressure that might result in speciation? Okay. So we have variations in weather, different predators and prey, different types of trees in the habitat, or all of the above. All right, all of the above, exactly. So all of those are different selection pressures, okay, that may result in speciation occurring in this population of birds. Right, good. Okay, number 10. So which of the following are basic causes of evolution? Okay, again, we have natural selection, we have genetic drift, we have mutations, or all of the above. All right, all of the above, perfect. Good. So all those can cause evolution as well as migration and non-random mating. And then the last one, which of the following can be used as evidence of evolution? We have the fossil record, analysis of similar or differing anatomy, we have DNA analysis, or all of the above. Right. Exactly. So all the above. Perfect. So all these can be used as evidence of DNA. Okay. So that was the homework. Are there any questions on the homework? No? Okay. So if there's no questions, like I said, if you were not able to do it before, you should hopefully be able to see it in your Canvas. So make sure you get that done by the end of the week. Sorry for the technical issues there. One thing I do want to kind of remind you guys and bring to your attention is that obviously next Tuesday, so a week from today, we're going to have our final exam. So to help you study for it, you can kind of go through the homework, you can go through the previous exams. And then there is also this practice final too. Okay, it's a very short practice final, about 30 some questions, I believe. So you can use this to help you kind of get an indication of what types of questions you will see on the final. Okay. So make sure you go through this. There's a final practice final, and then there's a key as well. So make sure you take a look at that. And yeah, you can use that to study. Oh, I forgot to upload the previous exams. So I will upload the previous exams onto Canvas later today, so you can use those to study. My apologies for that. I meant to do that last week and I forgot. So Alan's question is the final going to be like the other 2 exams online? And yes, it will be. So it's going to be all multiple choice. It's going to be 75 multiple choice questions. The first 40 are going to come from the new material, so from chapters 11 and chapter 19. And let me go ahead and just write that here. So for the final, there will be 75 multiple choice questions. Numbers 1 through 40 are coming from chapter 11 and 19. Okay. Numbers 41 through 75 will come from the previous exams. Okay, they will come directly from the previous exams. Okay. So like I said, you can use those to help you study. Like I said, I'll upload those later today, and they'll be up here on the exam study guides section. And so you can use that to help you kind of prepare for that. Cool. And then there'll also be a couple extra credit questions too. Cool. All right. Any other questions regarding the final? Okay. So again, this will be next Wednesday, or sorry, next Tuesday. So I'll open it up at 8 o'clock in the morning, and then you will have until 11:59 p.m. to complete it. Okay. There's no time limit, so again, take as much time as you need, but make sure it is completed by 11:59. Otherwise, you're going to get a 0, and this is worth 150 points. Okay. So this is not something you want to not do, obviously. Okay. Okay. Any other questions on the final? Okay, so that is that. All right, so that is what's going on with the homework, or sorry, with the exams. So Paul's question is, what are we doing on Thursday next week? Nothing. So this is our last week of material, and we're gonna have our labs this week, and after this week, we just have the final, and then that's it. Okay, we got nothing else. So Angelina's question is, do we need to check into class on next Tuesday? No. We're not going to be doing anything on Zoom anymore after this upcoming Thursday. Okay. So we'll have class this week, Tuesday. We have class on Thursday. Next week, we won't be on Zoom. We don't need to check into class. We don't need to go into lab or anything. All we're doing is just taking the final. Once you take the final, you are free for the semester from biotech. Alan's question is there another homework? No, this is it. That was the last homework from chapter 11. The way that you can kind of use to prepare for chapter 19 is to go through the questions on the practice final. And so the practice final is going to have questions from, I believe, all the chapters, including chapter 19. So you can use that to help you study for what you should see or what you should expect to see on the final. Cool. Any questions? Okay. Any other questions? So Josie's question is there another lab? All we have is just a lab for this week. Okay. After this week's lab, there is no more labs. Okay. So this is it after this week. Okay. So we'll have lab this week, nothing after this week. All right, no problem. Any other questions? Victor, I'll have to check. I might have gotten it. I don't remember for sure. But I'll check for it and then I'll get back to you on that. Okay. All right. Any other questions? Okay. So if there's no questions, we'll go ahead and dive into today's material. Okay. So is everyone able to see the PowerPoint? Yeah, okay. All right, cool. So this is our last chapter that we're going to be going over. And so this is going to be chapter 19, and we're going to be looking at population demographics and dynamics. Okay? We're going to essentially be looking at how populations are going to be changing over time. So last week we looked at how populations can change in terms of kind of the genetic structure and the amount of alleles and genes that are inside of the population. We were looking at the effects of natural selection. We were looking at the effects of mutations and migrations and genetic drifts and non-random mating, and looking at how the population is going to change kind of in its makeup over a period of time. Okay. What we're going to be doing with this chapter is we're going to be looking at how populations are going to be changing in size. Okay. And so as we saw last week, populations are never going to be static. They're always going to be changing. There's going to be always some of the, at least one factor that's altering what a population is going to be doing. Okay, there might be, like we said, there might be natural selection occurring in a population. We might have migration coming into a population. We might have individuals leaving a population. We can have mutations popping up. We can have, non-random mating. We can have random mating. We can have all kinds of different factors that are going to lead to populations changing. Okay? So populations are never just static. They're never just unchanging. Okay? Populations are always kind of dynamic. they're always going to be getting altered due to a number of different factors. Okay? And so as population ecologists and population biologists, one of the things we want to be able to do is study how the population is typically going to be changing in size, okay? And how it's going to be changing kind of in its makeup in terms of the individuals in it. Okay? And so the statistical study of how we're able to look at populations changing in size is called demography. And that's more of it, like looking at the populations from a statistics standpoint and looking at how many individuals are being born, how many individuals are dying, how many individuals are in this particular age group, and so on and so forth. And you want to be able to look at all of the different numbers of a population and be able to understand what's specifically going on in that population that's causing it to change in its size. Okay, sometimes populations are going to be growing, sometimes they're going to be shrinking, sometimes they're going to be pretty static. But there's always going to be some factor at play that's going to cause those particular changes to occur. Okay. So that's called population demography. And then we can also use another tool called life tables. And so life tables will help determine the life expectancy of individuals at each particular age group, okay, within a population. And so we'll take a look at how that looks like in a second. Okay. In order to really study populations, the first thing we want to do is we want to understand the population size. Okay? Population size is just the number of individuals inside that population. So for example, if we have, if we're looking at Gilroy, there might be something like 30,000 people living in Gilroy, okay, or something like that. If we're looking at the number of squirrels on Gilroy's campus or Gavilan's campus, then there might be 200 squirrels or 300 squirrels or something like that. Okay? So that's just the number of individuals inside that population. Okay? Now another important kind of number that we want to be paying attention to is the population density. Okay? So population density is the number of individuals that exist per unit area. That's going to be how densely packed your populations are. Okay? So for example, if we have more individuals in a smaller space, that's going to be a higher density. More individuals in an area, okay, that's going to give us a higher population density. Okay? If we have individuals that are more spread out in the population, okay, then the population is not going to be as dense. Its density is going to be much more reduced. Okay? And so here we see a little bit of a graph here. And so looking at this graph, what do we notice about a general trend? Here we're looking at the mass of different organisms and then the density, the population density of that particular population. What do we notice here? Okay, based on our graph. What general trend are we seeing here? So Josie's saying the bandicoot are closer together. Yeah, but I'm talking just across the species. Across the species, what's the kind of overall general trend here in terms of the mass and the density? So the denser so Paul says the denser the population, the more mass. Reverse that. So basically, the larger the animal is, the more mass that particular animal has, okay, the less dense that population is going to have. Okay, so this is a logarithmic scale. I think that probably confused some people. So this is on a logarithmic scale. But essentially what we see here is as the mass increases of that particular organism, okay, then the population density has to be smaller. Okay? They cannot exist in very large populations. Okay, so for example, we're looking at a small sized animal like we'll just say like an ant, for example, right? So ants, they're very, very small organisms. Okay, they have a small mass, but they can have a very high density. Okay, they can have a high population density. You can have lots of ants in a very small space because there's going to be enough resources to sustain them. Okay. However, if you try to take a bunch of bears and you put a bunch of bears into a small space, okay, that's not going to work because bears are going to require a certain amount of resources in order to sustain them. Okay? So bears, because they're much larger animals, they have to be more spread out. They cannot be in as dense populations. Okay? And so that's what we see here. Okay? So basically, the more density a population has, the higher the population density, the more competition there's going to be for resources. Okay? So if we have a larger animal, for example, larger animals are going to need a certain amount of food in order to sustain them, and then a certain number of mates, right? So if we have a larger population density, okay, if you have a larger population density, we're going to end up having more competition for those resources because there's just more individuals there that need access to those resources. If you have a smaller population density, the competition isn't going to be as great because there's going to be much more spread, I guess, much more individuals spread out in the population. Okay. Does that make sense? Yeah. Any questions on this idea here? Okay. All right, so that's that. So that's what we see here. So smaller animals can have a higher population density than larger animals do. Okay? Large animals cannot have a high population density because, again, there's not enough resource to sustain that many large organisms in a small space. Okay. All right, so what we want to do is, as population ecologists, is we want to be able to estimate the population size, okay? We want to be able to determine how many individuals there are in a population. Now, there's a couple ways to do this. The most accurate way is to go out into the population and just count everything, count all the individuals that are present there, okay? That's the most accurate way, and you're going to get a pretty accurate number, but usually this isn't really the most realistic or the most feasible way of doing things, okay? So, for example, if we wanted to go and count the number of redwood trees inside the redwood forest, we could do it. You know, we could actually go out and count all the redwood trees, okay? But it's not realistic, it's not feasible to do because there's a whole lot of factors we got to account for. We got to make sure that we're, actually counting all of the redwood trees, which would take forever to do. We would have to make sure we're not double counting. We'd have to make sure that we have some way of knowing which ones we counted and which ones we haven't counted. We got to make sure we didn't miss any. And then, there's a whole lot of factors that we got to account for. If we try to do the same thing with squirrels, and if we wanted to count all the squirrels on Gavilan's campus, again, we could go out and do it, okay, but there's no way of knowing, did we count all the squirrels, okay? Were there some squirrels that we didn't count that were hiding in trees? Were there some squirrels that were hiding underground? Are there some baby squirrels that were just being born that we didn't see? There's a number of different factors at play that can, affect our ability to count all of these individual organisms, okay? So you can do it, like I said. It's just very, very, very difficult to do, and it's not really realistic. Okay, so instead we need some kind of way to kind of estimate populations without necessarily counting every single individual. Okay? And so what population ecologists do is they actually will go out into a sample population, okay, and then they'll estimate using different techniques and estimate the population size. Okay? And so based on the different organisms you're looking at, there's going to be different techniques that are going to allow you to be able to estimate the overall population size. Okay. So we're going to look at a few of these techniques. The first one is using quadrats. Okay. So quadrats are typically used for immobile organisms like plants or things that are not really going to be moving around a lot, like starfish and things like that. Okay. And so basically what a quadrat is it's usually going to be a square kind of shape here. And you can make this square out of either wood or like PVC pipes or metal or whatever you may use. And you spread these quadrats over your sample population. Okay. Once you kind of take your quadrat, you spread it over your population, you've got to put it in a number of different areas throughout your population. And you're going to count the number of individuals that you see inside your square, okay, inside your quadrat. Okay. So for example, you can kind of orient them in random quadrats, random locations, or you can do them in a systematic manner. It just kind of depends on what you want to do. But basically, let's say we did one quadrat here, and we found, let me do this in blue so we can see this. Let's say we're looking for, we want to try to estimate the size of the population of California poppies, okay, in Gilroy, for example. Okay. So let's say over here in this field, we were able to find 5 California poppies in this quadrat. Okay. Let's say another one, we were able to find another 5 here. Okay. Let's say this one, we were able to find another 5 here. Let's say this one, for example, we were able to find 15 poppies. This one had another 15 poppies, and this one had another 15 poppies. Okay. So we spread your quadrat, you find out how many are inside each individual quadrat. Let's say this is 1 square, each one of these quadrats is 1 square yard. Okay. And so once you kind of estimate how many is inside each individual quadrat, you now take an average and you say in 1 square yard, whoops, let me do this way. So let's say in 1 square yard, we had an average of 10 poppies. Okay. We had 5, 5, and 5, 15, 15, 15. Okay, so we had an average of 10 poppies per square yard. Okay. Let's say this entire this entire field here is let's say it's 10 square yards in total, or let's say it's 20 square yards. Okay, so let's say this is 20 square yards total. Oops, or 20 square yards total. So 20 square yards total. If we found that in each individual square yard, there's about 10 poppies. If you have 20 square yards, all you do is just multiply 10, which was your average, times 20, which was your total square yardage. And that would estimate your population to be about 200 poppies. Okay. So basically you take an average of the amount of individuals you find in each quadrat. Okay, you multiply that by the size of your entire population. Okay, and then you estimate your population size from there. Okay, does this make sense? There's a little bit of a kind of confusing way of going about it, but basically you take the average, okay, and then you kind of extrapolate that to the rest of your entire population size. Okay? So basically you do this enough times in order to get yourself an accurate estimate. Okay? And then from there, you can kind of determine, the average population size for that particular habitat or the entire population as a whole. Okay, just depending on what you're looking for. Okay. And so the number of quadrats that you use, the size of the quadrats, those are all going to be based on the type of habitat you're looking at, the type of organism you're looking at, and the distribution of those organisms throughout that particular habitat. Okay. So this is one technique that you can use for small populations. Okay. Any questions on how to use quadrats? Okay. Again, you just kind of lay them out in random locations, take an average, okay, and then use your average to extrapolate, what the overall population size would be for the entire population. Okay. All right, so that's that. Okay, so those are quadrats. Those are used for immobile organisms like plants. Okay. Now, if we were looking for mobile organisms like squirrels or things like that, okay, then we're going to use another technique called mark recapture. Okay, quadrats are not going to be useful for this technique because you have organisms moving from location to location. Okay. So instead, what we're going to do is we're going to use a technique called mark and recapture. Okay, so basically with mark and recapture, what we do is we go into a population and we capture as many individuals as we can. Okay, we capture as many individuals of that particular population that we're looking for. Okay, we capture them and then we mark them with some kind of marking. Okay, and then we release them back into the environment. Okay, now the mark that we use cannot be something that affects that animal's ability to survive. Okay, if it affects the animal's ability to survive, that's going to cause the population size to shift. And then we may end up, killing some organisms when we were trying to just estimate the population size. And now we're going to get different numbers from what we should have. Okay? So we want to make sure that the mark does not affect the animal's fitness. Okay? So you go out, you catch them, you mark them, and then you release them all back into the environment. Okay? Once you release them, you give them a chance to kind of mix back in with the entire population, mix back in with the habitat. And then maybe you come back to that population about a week, about a month, maybe a few weeks later or a year later, whatever you want to do. But you come back after a second time, and you catch as many individuals as you're able to catch. Okay. So once you capture them, you then determine the number that were already marked from the previous time you went out. Okay. Based on the number that you caught and marked the first time, the number that you caught the second time, and the number that were marked in that second catch, those three are going to be able to help you find or estimate the population size as well. And I'll show you a formula that you're going to use on the next page, on the next slide. Okay. So typically, if we have a larger population, then the chances of us seeing individuals that we already marked is going to be lower. Okay. Which makes sense, right? If you have a large population, the chances of you being able to, pick out one that was already marked is going to be relatively low compared to if you had a small population. If you have a small population, then the chance of you being able to see a marked individual is going to be relatively high because you're working with a smaller pool. Okay. So this is the formula here. Okay, so the formula is you take the number that you marked in the first catch, the total number that you caught and marked in the first catch, you multiply that by the total number that you caught in the second catch, and you divide that by the number that were already marked, okay, that were inside that second catch. Okay. And that will give you an estimate of the population size. Okay. So we'll do an example on the next slide here. But these are some different types of markers that you can use. Okay, so one marker that you can use is to give them a color. Okay. On some organisms, you can give them like a tattoo on their upper lip. Okay. Other ones like birds, you can give them like a little tag on their talons. Okay, for smaller mice, you can give just a little Sharpie on their belly. Okay, so all of these different techniques can be used to mark the individual, but they're not going to affect that organism's ability to survive. Okay, so these would be good markings. Okay. So here we have an example. Okay, so here we have in a field you capture and mark 50 squirrels. Okay, and then you release them back into their habitat. Okay, a week later you return and capture 30 squirrels. Of those 30 squirrels, 10 are already marked. Okay, so estimate the size of this squirrel population. Okay, so go ahead and use your phone's as calculators, or get a calculator and try to work this one out and see what you come up with. Okay, and then let me know what you get. So we have 17, we have 100, 150, or 1,500 squirrels. All right, so Alan says C. Yeah. Anyone else? Can anyone confirm or deny his answer? Anybody else? Anybody else? Yeah, Kimberly says C as well, and C is correct. Exactly. Oh, this is perfect. So again, what you do is you take the number that you caught in the first catch, which was 50. Okay. You multiply that by the number that you caught in the second catch, which was 30 squirrels. Okay. And then you divide that by the number that were already marked in that second catch, which is 10. Okay. So you 50 times 30, which gives you 1,500. Okay, divide that by 10, and you get 150 squirrels. Okay, so we know our population size is about 150 squirrels. Okay. So this is one formula that you will need to be comfortable with for the exam. And I'll give you all these formulas on the exam as well. So we're going to go through a few that you will need to be familiar with and know how to use. Okay. So this is one of them. Okay. So know the mark and recapture formula. Okay. All right, good. Okay. So that's mark and recapture. Any questions on the two kind of techniques to use in terms of estimating population size? Any questions so far? No. Okay. Okay, so the next thing that we're going to be taking a look at is species distribution. Okay, this gives us an indication of the density of our populations. Okay? And so species distribution patterns are just the patterns of how those particular organisms are going to be spread and distributed throughout their population. And we've got three different types of species distribution patterns. Okay, we have uniform distribution, we have random dispersion, and then we also have clump dispersions as well. Okay. So depending on the pattern that we're looking at, those are going to influence the calculations that population ecologists are going to use to help estimate their population size. Okay, we're not going to worry about that for the sake of our class, but if you were taking it like a population ecology class, these would be some things that you would have to take into consideration. Okay. And so these also give an indication of how those organisms interact with one another inside their populations. Okay. So we're going to start by taking a look at random dispersions. Okay. So random dispersions is where we have individuals that get randomly placed throughout the environment. Okay, so these will typically take the form of wind dispersed seeds. Okay. So any plant that has wind dispersed seeds, like dandelions, okay, the wind is just going to just pick up those seeds and then disperse them throughout a field, throughout a location, throughout an environment or a habitat. And wherever those seeds land is going to be where they sprout new dandelions. Okay, so there's no rhyme or reason to where these individuals pop up. Okay, they show up randomly. throughout the population, as long as they land in a favorable environment, that allows them to survive. So we have individuals that are placed randomly throughout the habitat. That's random. Next one is clumped. And so clumped distribution is where we have individuals that will exist, usually in some social groups or that live in herds and things like that. Okay? So for example, here we've got elephants, for example, that usually will travel in herds. Okay? So this would be an example of a clumped distribution. Okay? We can have some that are like schools of fish, which are going to also just as a social group. That'd be another clump or another type of a clump distribution there. Okay. And plants, we can also see clump distributions where if you have a tree that drops its seeds directly below it, okay, as long as those seeds are able to, take root and then sprout up, and now you're gonna end up with clumps of different trees in different locations too. Okay. So that's another way we can end up with clump distribution in terms of plants. Okay. So clump distributions are where we have individuals that exist in groups. Okay. Okay. And again, that kind of gives an indication of how those organisms will exist with one another and how they're going to interact with one another. Okay. Okay. The next one is uniform distribution. This is where individuals are evenly spaced out throughout the habitat. Okay. So this is typically what we see in territorial animals. So for example, when we're looking at penguins that are defending their nests, they all need to stay in a particular zone. Okay. Each penguin is going to have its own nest, and it's going to protect the area immediately surrounding that nest. Okay. If one penguin tries to enter, for example, penguin A tries to enter penguin B's environment or penguin B's, location, okay, or territory, okay, penguin A better be ready to catch these fins. Okay, they're gonna start getting feisty, and they're gonna start fighting. Okay, so they're gonna be kind of evenly spread out, and they're gonna keep their own distance from one another in order to kind of protect their own nests, but also kind of defend their offspring as well. Okay, so that's gonna be one place we're gonna be able to see uniform distribution, where you have organisms that are evenly spread out. Okay. Another example would be some plants, and so there's certain plants that are able to secrete different toxins into the surrounding area, and so when they secrete those toxins, they kill everything within, let's say, a 12-inch radius. Okay, so anything within that 12-inch radius is not going to be able to survive around that plant. And so that's going to cause this plant to have, a specific location, and they're all going to be evenly spread out as they all produce those same toxins. Okay. So that's a uniform distribution. Okay. So those are the three types of distribution patterns. Okay. A question here. So we have wolves often travel in packs. Which distribution pattern do wolves typically exhibit? Is this going to be random distribution, clump distribution, or uniform distribution? What do we think? So Jackie says B, Kimberly says clumped as well. Yes, and that is true, exactly. So wolves traveling in packs, they're gonna be in groups, okay? And so this would be an example of clumped distribution. Perfect. All right, good. Okay, so as we continue to go through our population size and as we start to understand and study our population size, what we're gonna be doing is we're gonna be looking at the demography of that particular population. Okay, we want to be able to study how the population is going to be growing in size over a period of time. Okay. The three most important factors to help us determine that is the birth rates in that population, the death rates, as well as the life expectancies. Okay. And so the easiest way to kind of look at this is using some life tables and some survivorship graphs. And we'll take a look at these right now. Okay. So the first ones that we're going to take a look at are life tables. And so life tables will give information about the life expectancy of an organism at different stages of its life, okay, different stages or different ages, I should say. Okay. And so a life table will include kind of many characteristics, but of those, the most important ones are going to be the probability of individuals of each age group dying before its next birthday. Okay. It's going to give us the percentage of individuals that will die at that particular age group, which is the mortality rate. And then the life expectancy at each individual age interval as well. Okay. So all three of those factors are going to help us to be able to understand what's going to be happening and kind of predict what will happen inside of a population. Okay. So this is what life tables will look like here. Okay. So this is what a life table looks like. And so in a life table, you're going to have 5 columns typically. The first column is the age of that particular organism. Okay. So here we have, from 0 to 0.5 years, half a year to 1 year, 1 to 2, 2 to 3, 3 to 4, and so on and so forth. Okay. So we have the age interval in column A. I'll get to column B in a second. Column C is the number of individuals that survive at the beginning of that age group. Okay. Column B is the number of individuals of that particular age group that are dying. Okay. So the number of individuals of that surviving group that die, okay, during the course of that year. D tells us the mortality rate based on these 2 factors here. And then column E gives us the life expectancy, or the amount of time we can expect that particular organism to survive, okay, typically. Okay. And so by using this life table, one of the things that we want to be able to calculate is the mortality rate. And this is another formula I want you to be comfortable with for the exam too. So in order to calculate the mortality rate, you take the number of individuals that are dying, okay, within an age group, and you divide that by the number of individuals that survived at the beginning of that age group. Okay. So for example, and you multiply that by 1,000. Okay, this is usually going to be reported in individuals per 1,000. So here we're looking at some Dall mountain sheep, and if we look at the first half of the year that they're born, there's going to be 1,000 individuals that are born. Okay. Of those 1,000 individuals, 54 die. Okay. So we do 54 divided by 1,000, okay, and then you multiply that by 1,000, which gives us 54. So we can expect the mortality rate to be 54 sheep per 1,000 that will die. Okay. So then if you go to the next table, then if 54 died, then the next surviving group is going to have a total of 946 individuals. Okay. And we can expect 145 individuals to die out of this 946. Okay. So if we do 146 divided by 145 divided by 946, again, multiply that by 1,000, the mortality rate becomes 153.3 sheep per 1,000 will be expected to die. That's the mortality rate, and so on and so forth. Okay. So this is how you would be able to calculate the life expectancy at each individual age range. Okay. So question here. So in the Dall sheep, what is the mortality rate of sheep between the 6 to 7 years of age? Okay. Again, go ahead and take a calculator and see if you can work that one out, and see if you can figure this one out here. Okay. What would the mortality rate be? Anyone have an idea for this one? So Kimberly got 69.8, and that is correct, right? So in order to do that, what you would do is you would take the number of individuals that survived, or you're looking at these 2 individuals here, the number that die at that particular age group, and you divide that by the number of individuals that survived at the beginning of that age interval. So you have 48 sheep that died out of 688 that survived at the beginning of that. Multiply that by 100, and you end up with about 69.8 sheep per 1,000. And that would be the mortality rate between the ages of 6 and 7. Okay. So remember, that's another one that you will need to be comfortable with for the exam as well. Okay. Calculating mortality rate. Cool. All right. Any questions so far? Okay, cool. Okay, so that's what we see in a life table. Okay. We can also look at survivorship curves. And so survivorship curves are these graphs that will give us an indication of the number of individuals that will survive at each individual age group. Okay. So it's almost like a life table, but it kind of gives us different types of information. Okay. And so this allows us to be able to compare what's happening at different life stages between various organisms. Okay. So we have type 1 curves, we have type 2 curves, and then we have type 3 curves, as we see down here. Okay, so we'll take a look at all 3, and we'll start with the type 1 curves. So our survivorship curves are going to have time on the x-axis, which is going to give us an indication of how long how long of a time period is occurring. And on the y-axis, this gives us the number of individuals that are surviving. Okay. So what we see here in this type 1 survivorship curves is that during the early and the middle years of life, survivorship is high. Okay, there's a lot of individuals that are able to survive during the early and middle years, and which tells us that mortality is going to be relatively low. Okay, the mortality rate is going to be low for these individuals. Okay. However, the mortality does increase later on in life. Okay, so as we get to later on in life here, we can obviously see that survivorship is going to decline very drastically. Okay. And so this tells us that individuals who exhibit type 1 curves are individuals that produce very few offspring, but provide very high levels of care. Okay? They provide lots of care to their offspring, which allows them to be able to survive during the early and middle years. Okay? And then as they get older, they just kind of get old and then they die. Okay? Basically, as we have individuals being born, there's going to be lots of care, lots of parental care, and that's going to improve the survival of their offspring. Okay, so this is going to be what we find in humans and most other mammals. Okay. We have a typically very high chance of survival early on in life, and then as we get older, the chance of us dying, obviously escalates. Okay. So those are type 1 curves. Okay. Type 2 curves are where you have mortality that's pretty constant across the board. Okay. So from one age to the next, the likelihood of dying is about the same. Okay. And so this is what we see in birds. Okay. And so birds, whether they're firstborn to later on in life, their likelihood of survival or their mortality rate is going to be pretty much constant. Okay. So constant mortality. And then the third one are type 3 curves. And so type 3 curves are where you have a high mortality rate very early on in life, but then as you start to pass through a certain age range, okay, if you're able to make it through that particular age range, then the likelihood of surviving becomes pretty high, and you pretty much just stay relatively high survivorship there. Okay, so that's what we see with trees, for example. Okay, so trees will have a pretty low or a pretty high mortality rate early on in their life. If we see like seeds that are getting planted right away, the chance of them surviving is going to be pretty low because of predation or because they didn't land in a favorable environment. There's going to be lots of factors at play that are going to cause those plants to not be able to survive as well. Okay? But if they are able to take root and if they are able to survive to a particular age range, the likelihood of them surviving is going to be relatively high, and they're going to be able to survive for long periods of time, even deep into their advanced years. Okay? So this is what we see in organisms that produce lots of offspring and don't provide them with a lot of care. Okay? So that's going to be trees. Obviously, they're not going to be able to provide any protection or any kind of care for their offspring. And then like marine animals and marine invertebrates as well. So like fish, I'm trying to think of what else, like starfish, things like that. Anything that's in the ocean, any kind of marine invertebrate, those are typically going to produce lots of offspring, and they're not really going to provide any parental care because they're going to be pretty self-sufficient from the time they're born. Okay. So those are type 3 curves. Okay, so that's that. Any questions so far on anything we've looked at going forward here? No. Okay. All right, so that's going to lead us into chapter 19.2, which is about population growth and regulation. Okay. So with population growth, one of the things that we're going to be able to see is populations either growing or declining over a period of time. Okay. And so again, one thing that we want to do as population ecologists is we want to be able to understand how quickly our population is either growing or how quickly it's declining. Okay? And there's going to be different ways that we're going to be able to do that. Okay? And again, that's going to be done over time. So there's 2 different types of population growth. The first type is called exponential growth, okay, where you have no limit on the population growth. Okay, it's just going to continue to escalate and grow without any limit on its growth. The other type of population growth is called logistic growth. And so this is where you have some kind of a limit on how many individuals are able to survive in that particular environment. Okay? There's a limit to your reproductive growth as the population size begins to increase. Okay? So as we get to a limit, okay, you're not going to be able to pass that particular limit when you look at your population size. And so we're going to take a look at those 2 different characteristics or different types of population growth. And we're going to start with exponential growth. Okay, so exponential growth is basically, like we said, it's growth where you have the population size which increases very rapidly whenever you have resources that are abundant. Okay? Whenever there's lots of resources available, you're able to just produce offspring and produce offspring and produce offspring really without very much limit, okay? Without any limit really. Okay? And so population continues to grow kind of without a limit here. And so in order to calculate the growth rate, and again, this is one formula that you also need to be comfortable with. I listed all the formulas in red. And so the one that you want to be comfortable with for growth rate in terms of exponential growth is just by subtracting the birth rate minus the death rate, okay, during that particular time interval. Okay? So we take the birth rate minus the death rate, that will give us the growth rate of the population. Okay? Now this growth rate can either be positive, okay, if you have a birth rate that's larger than the death rate. Okay? So if the birth rate is larger than the death rate, your growth rate is going to be positive, which means your population is going to be growing. Okay? If your death rate, excuse me, if your death rate is larger than your population, or than your birth rate, okay, let's see, if your death rate is larger than your birth rate, then the growth rate is going to be negative, and so that's going to be an indication that your population is going to be shrinking. Or if they're the same, if the birth rate is equal to the death rate, they're going to be, your growth rate is going to be 0 here, okay? And so your population is not going to be growing during that time period. Okay? So this is going to be an indication of how your population is going to be growing. Okay? Now, in order to determine the number of individuals that are actually being added to the population or removed from the population, what you do is you take the growth rate and you multiply it by the population size. Okay? And that will tell you the number of individuals that are either being added to the population, either by birth or the number of individuals that are being removed by death. Okay? And again, N refers to the number of individuals in the population as a whole. So again, growth rate is the birth rate minus the death rate. Population growth, in terms of the number of individuals being added into the population or removed, is going to be the growth rate times the population size. Okay, so an example of exponential growth is what we see in bacteria. And so if you kind of go back to chapter 6, bacteria, when they replicate, are going to replicate using binary fission. Okay? And so you have one bacteria that eventually splits into two. Okay? So let's say we're doing some kind of experiment and we want to look at how this population of bacteria is going to be growing. And we start off with 1,000 bacteria at time 0, okay, at our initial moment. Okay, so at time 0, okay, we have 1,000 bacteria. After an hour, okay, our 1,000 cells have divided into 2,000 cells, okay, through binary fission, and that's what we see here. Okay, then after hour 2, okay, now again they double again. Okay, now we have 4,000 individuals. Okay, at the third hour they double again. Okay, now we have 8,000. Hour 4 they double again. Now we have 16,000. Hour 5 they double again. Now we have 32,000. Okay, so not only are we seeing the population growth increase in and of itself, but we're also seeing the growth rate also increase. Okay? We went from a 1,000 to 1,000 to 2,000 bacteria. Okay, that's a difference of 1,000 during the first hour. During the second hour, we increased by 2,000 bacteria, from 2,000 to 4,000. Okay, during the third hour, we increased from 4,000 to 8,000, so that's another increase in 8,000 or 4,000 bacteria. Okay? Then from 8,000 to 16,000, that's a growth rate again. It's also increased, and then from 16,000 to 32,000 within the next hour. So we can see that the population is growing with a much faster growth rate, okay, over a period of time. Okay? That's typically what we'll see during exponential growth. Okay? Not only is your population growth increasing, but the growth rate itself is also increasing over that period of time too. Okay? So that's what we see with exponential growth. Okay? Now, the more kind of realistic type of growth pattern that we'll typically see is called logistic growth. Okay? So logistic growth is kind of more representative of what we actually see in the real world. And this indicates that we have limited resources. Okay? Limited resources in that particular habitat or in that environment. Okay? And so what we end up seeing here is we see the growth rate beginning to increase over a period of time. Okay? Then it begins to slow until it reaches a point that's called the carrying capacity. Carrying capacity, also referred to as the K value. Okay? And so the carrying capacity is the maximum population size that habitat can sustain, or that environment can sustain, due to the resources that are limited in that particular area. Okay, so if we have limited resources here, the population is not going to be able to go beyond the carrying capacity. Okay. Now, question for you guys: what do you think will happen if our population size drifts above the carrying capacity? Okay. If let's say for example here, let's say for example, we start drifting above our carrying capacity. Let me draw that again. We drift above our carrying capacity. Okay. What do we think will happen if our population size drifts above the carrying capacity? Okay. Anyone have an idea of what we should expect to see? So Kimberly says there won't be enough homes, food, water, etc. Exactly. You don't have enough resources in order to sustain the number of individuals. And so what do we think will happen? Okay, what's going to happen to the population size then? Anyone have an idea? Okay, it'll decline, exactly. So the population size will decline back down to below the carrying capacity. And so it'll drift back down here. Okay. So that's what we should expect to see. Okay, and the reason for that is because there's not enough resources available. Okay, so because there's limited resources, okay, and there's not enough resources to sustain that many individuals, the death rate is going to overwhelm the birth rate. Okay? Because there's not all the individuals in the population are going to be able to access those resources. They're not going to be able to get enough food. They're not going to be able to get enough water. They're going to end up dying. Okay? And so as they die, the death rate is going to increase. And so the population is going to drift back down to below the carrying capacity. Okay? So that's typically what we saw with this example of seals inside this time period here. Okay? So there's a population of seals, starts to drift above carrying capacity. As it starts to drift above, there was no longer enough food for them. Okay? And so as a result, a lot of seals ended up dying because they weren't able to access enough resources, and the population size drifted back down below carrying capacity, which is what that particular environment is able to sustain. Okay. Okay. This makes sense so far. Any questions? Okay. Cool. All right, good. Okay, so in order to understand carrying capacity, there's another way that we're going to be able to determine the number of individuals, okay, being added to a population before you reach carrying capacity. Okay? And so what you can do is you take the growth rate times the population size, and then you multiply that by the carrying capacity subtracted by the population size divided by the carrying capacity again. Okay? And so this will give you the number of individuals that you can add into the population before you reach carrying capacity. Okay. So again, this is another formula to be comfortable with. And like I said, you'll have all of these on the exam that you can use. Okay. So this is what the graph for logistic growth looks like. So we're going to kind of piece this apart and kind of explain what's going on in each different segment of this graph. Okay? So the first part, we're going to look at this green region here. And so during this green region, we have a small population size. Okay? The population is small. Okay? We have lots of resources available. Okay? Because there's so many resources available, because we have such abundant resources and such small number of individuals, the population is able to grow very quickly because there's lots of resources for survival here. Okay? So we have a very high growth rate during this period. Okay? As the population size begins to increase, okay, resources are now becoming slightly more limited. Okay? And so the population's growth rate is going to begin to decline over this period of time here. Okay. So population is increasing, resources are becoming a little bit more limited. Okay. Then at the last portion, this top orange portion here, the population is extremely large. And so at this point, you start to approach your carrying capacity because your resources are now becoming extremely limited. Okay. So because resources are extremely limited, the population size now reaches its maximum. okay, which is at the carrying capacity. Okay. So that's typically what's happening at different points along this graphic. Okay. Again, the first graph, or this first segment, we have a small population size, we have lots of resources available, so we have a very high growth rate. Okay. In this middle region, the population size begins to increase. As you have more individuals in that population, they're going to be utilizing more of those resources, and so the resources become a little bit more limited. Okay. And so the growth rate begins to slow. Okay. Then as you reach the top portion, this orange region here, the population is now extremely large, and so resources become extremely limited because there's so many individuals using those resources. Okay, so we reach a maximum on our population size. All right, cool. Okay, so logistic growth kind of tells us, and kind of is an indication that there's lots of phenotypic variation inside within the organisms of that population. Okay? That variation is going to allow some organisms to be able to survive better than other ones do. Okay, and so the ones that are able to survive better are the ones that are able to outcompete other individuals for access to those resources. Okay? And so that's what we refer to as intraspecific competition. Okay, so intraspecific competition is competition between individuals of the same species for access to resources. Okay. Okay. So again, that's going to be due to variation. Different individuals will have different traits that will allow them to be able to access those resources better than others. Okay. So that is logistic growth there, and that's intraspecific competition. Okay. So based on what we just talked about, where would intraspecific competition most likely be seen? Would it be most likely seen in the orange region, the red region, or the green region of this graph? Where would we see the most competition? So Josie says the orange region, Kimberly says the red region. Anyone else? One of these is correct. Let's see if we can break this tie. Where would we see the most competition? Anybody else? So Yetzi says orange. Oh, Jackie says red. Julio says orange too. So we had about a 3-2, and so this would be in the orange region, right? So where we have the most individuals, yeah, sorry, Kimberly. Where we have the most individuals is where we're going to see the most competition, right? So the more individuals we have there, okay, the fewer resources are going to be available, okay? And so there's going to be much more competition for access to those resources. Okay? Here in the red region, we still have a good number of resources available, okay? So there's not going to be as much competition because there's still lots of resources and not a whole lot of individuals present. Okay? Once we get to this orange region, there's going to be a lot of individuals and very few resources. So there's going to be lots of competition for access to those resources. Okay. Does that make sense? Okay. So that's why we see in the orange region, that's going to be the most likely location where we're going to see the most competition. Okay. All right, cool. Okay, so when we look at different species, we can classify them in two different ways in terms of their demographics. The first ones are what we refer to as K-selected species. So K-selected species are individuals that exist at usually closer to carrying capacity, which is kind of where the K comes from. And so they typically live in relatively stable environments. Okay? So K-selected species are individuals that will produce very few, pretty large offspring over the course of their lifespan, and they provide lots of parental care to their offspring. So these would be things like elephants. Humans are an example of K-selected species. Giraffes. Typically, pretty large animals are going to be more K-selected species. Okay? They produce few offspring, but they provide lots of care for their offspring. So they spend the majority of their energy, caring for the few offspring that they do have. Okay? The flip side of this is the R-selected species, and these are mostly based on their growth rate. Okay? So they're more adapted for unstable environments. And so these ones are offspring, or are individuals and populations that will produce large amounts of offspring, large amounts of very small offspring that are mostly self-sufficient, right at birth. Okay. So these would be things like jellyfish, plants, smaller organisms that produce very large amounts of offspring. And the reason for that is, again, because their offspring are going to be mostly self-sufficient. So the parents are not going to be producing lots of parental care because the offspring can kind of survive on their own from the time they're born. Okay, so those are our selected species. Okay. Cockroaches too, yes. Cockroaches would be a big example of our selected species too. Yeah. Cool. Okay. As we look at population dynamics, one of the things that we're going to be looking at is how population sizes are going to be changing. And so what we can see is that carrying capacities are almost never going to be constant. Carrying capacity can change over time based on a number of different factors, a number of different events that are happening in that environment. Okay? So for example, the carrying capacity may be higher during the summer than it might be during the winter just because different resources are now being available than compared to others. And so for example, if there's more resources available during the summer, then the carrying capacity is going to be higher than during the winter when there's not as much resources available. Okay. We can see some natural events, natural disasters that can take place. And so when those disasters occur, they affect the number of resources that are available. If you cause the number of resources to decline, that's going to shift the carrying capacity downwards again as well. Okay. And then multiple populations are going to be able to compete for the same, or are going to have to compete for the same resources, even between species, okay, multiple species. And so, for example, here we see, a leopard kind of competing with a hyena for those resources. And so that's where we get interspecific species, where you have competition for the same resources across different species here. Okay. And again, that's going to be due to carrying capacity shifting as a result of resources being either declining usually. Okay. So we can have different types of factors here. So we can have density-dependent factors and then density-independent factors. Density-dependent factors are different factors that are going to affect population size based on the population density. Okay. This is going to affect population growth based on the density of the population and affect mortality as well. Okay. Density-independent factors are where we have factors that are going to affect the overall mortality rate, but they're going to affect the mortality rate for reasons other than the population density itself. Okay. So we're going to take a look at a few of these, and we're going to kind of speed through this. I know we're kind of getting to the end here. But we're going to look at density-dependent factors. So density-dependent factors can involve biological factors like predation. And so if we have predation, for example, if there's a low density of prey available, that's going to cause the mortality rate of the predator to increase as well. Okay? And that's going to be because the predators are not going to have access to as much resource. Okay? If there's not as much prey available, if there's not as much food available, then predators are going to end up dying because they're not going to be able to access their prey because the prey are dying too, okay? Or there's less of them available. Okay? Intra and interspecific competition can also factor into different density-dependent factors too. So if we have more competition, that's going to result in more individuals dying. And if we have more individuals dying, we're going to end up with lower reproductive rates, and that's going to cause the population growth to decline as well. And then parasites and disease, right? So if we have lots of individuals packed together in a very small space, it's going to be a lot easier for diseases to jump from one individual to the next and then cause mortality inside that population. So denser populations will typically have higher mortality rates, and that's usually going to be due to the presence of competition, okay, competition for those resources, and then the presence of diseases hopping from one individual to the next, and then causing death there too. Okay. Those are density-dependent factors. And so an example was in Australia, they were doing an experiment to see how population density affected the mortality rate in donkeys. And so they had two different populations. They had one population that had a high density of donkeys, a high population density of donkeys, and then there was another population that had a low population of density of donkeys. Okay. And so what they saw was that the high density population had a much higher death rate, a much higher mortality rate than the low density population. Okay, which makes sense because if you have a high density population, there's going to be more competition for resources, and the resources are going to become more scarce. Okay? If there's a scarcity in those resources, okay, if those resources are not as available, okay, that's going to cause individuals to die, and the mortality rate is going to be higher, okay? Compared to individuals where you have a lower density. Okay? If there's low density, there's not going to be as much competition for those resources, okay? And so mortality rate is going to be lower. Okay? And so one of the things that they saw was that in donkey mothers, there's a lot of malnutrition in the mothers in the high density populations because, again, they weren't able to access those resources. Okay? They weren't able to get as much food. Okay? And at the same time, that also led to juvenile mortality as well, because, again, they were not able to get as much nutrition from the mothers. Okay? So mortality rate declined, or mortality rate increased in mothers and in juveniles in these high density donkey populations. Okay, so those are density dependent factors. We also have density independent factors. Okay, so density independent factors are factors that will affect survival or increase mortality rate for reasons other than population density itself. Okay, so these are going to be things like physical factors like environmental changes. For example, we're looking at forest fires, earthquakes, hurricanes, volcano eruptions, things like that. Okay, those are going to affect an individual's chance for survival. Okay, so these are considered density independent because the ability to survive, forest fire, for example, okay, the ability of a deer to survive a forest fire is going to be the same regardless of the population density. Okay? If there's lots of deer available versus if there's a few deer present, okay, the survival chances are going to be the same. The only thing that's going to affect its ability to survive is how fast it's going to be able to get away from that fire, or how fast it's able to get away from that disaster. Okay, if it's able to get away, its chance for survival is going to be higher. If it's not able to run away fast enough, then it's not going to be able to survive. Okay? So the chances of survival are going to be the same regardless of the number of individuals that are present in that particular environment. Okay? The density is not going to impact whether an individual is going to survive or not. Okay? In the case of natural disasters and things like that, and environmental changes and so on and so forth. Okay? So for density independent factors, populations that have a higher population density are going to be able to rebound better from natural disasters than low population density. So this is where we kind of see the intertwining between density dependent factors and density independent factors. Okay? So for example, if we have this forest fire in a high density population of deer, okay, their deer population is going to be able to rebound a lot better after the forest fire because there's going to be more survivors, okay, than a low density population. Okay? In a low density population, you're going to have only a few individuals, so you're only going to have a few survivors, and it's going to be a little bit harder to get your population size back up to its original level. Okay? If you have a high density population, okay, then it's going to be a little easier because with more individuals, you're going to have more survivors. With more survivors, you're going to be able to get the number back up to the original level as it was previously. It's going to be able to help your population size recover a lot better, okay, with a higher density population. Okay. All right, so that's that. That is chapter 19.1 and 19.2. Sorry, we went a little bit long here. But are there any questions on these factors here, 19.1 and 19.2? Anything we talked about so far? Okay, so if there's no questions, you guys are free to go. The classwork is available on Canvas, so make sure you complete that. So get that done, and then the what else? Oh yeah, I think. And then if I see you for lab today, I will see you guys later. If not, I will see you guys on Thursday.